Yeah.
Well, good morning, everyone. Welcome to this webinar. We'll start here in a minute or two. It always takes people a while to get connected. My name is Tom Brum. I'm a professor in the Department of Agricultural and Biosystems Engineering here at Iowa State and a participant in the consortium work that we're doing at Iowa State and beyond. I see we come from a wide variety. It's 8 o'clock here in Iowa and also 8 a.m. in Honduras and all the way to East Africa where it's tea time in Kenya. So welcome everyone. In the effort of keeping us on time, I'm going to keep going. So just a reminder, the Consortium for Innovation in Post-Harvest Loss and Food Waste Reduction was created as a partnership between a number of universities and research institutions across the globe with the mission to improve livelihoods, preserve food nutrients, and realize an efficient food system. The consortium also focuses on building academic and entrepreneurial capacity by engaging researchers and students in multinational, multidisciplinary teams, together with experts and entrepreneurs from both the private and the public sectors. So this is great work that the consortium doing. I want to acknowledge the funding organizations because certainly without them, a lot of this work would not be possible. So the Rockefeller Foundation and the Foundation for Food and Agricultural Research are the main funders of our consortium. So the title of this session today is Update on Post-Harvest Loss Mitigation of Grains, Shea Nuts, and Other Perishable Crops. And we're doing some student updates from the work that we're conducting here at Iowa State. We have 3 speakers. The first speaker, I'll briefly introduce them. The first speaker is Jorge Chiquez. He's a PhD candidate in industrial and agricultural technology here at Iowa State. The next speaker is Christine Concepcion Ignacio. She's a PhD candidate in agricultural engineering here at Agricultural and Biosystems Engineering here at Iowa State. And then our third speaker is George Obenga-Kofi. He's a PhD student in agricultural and biosystems engineering, again, here at Iowa State. So thank you all 3 for getting up this morning and getting all coffeeed up so you're ready to speak later. So with that, without further ado, I'm going to ask Jorge to share his screen and give his presentation. He has about 10 minutes and then we'll have 4 minutes after his presentation. I'll be monitoring the chat. So if you have questions that you want to ask, you can enter them there. So with that, Jorge, you're on board.
Okay, thank you, Dr. Brum. Hello, everyone. And again, my name is Jorge Chiquez, and welcome to this presentation on identifying critical drivers of maize, mango, and tomato post-harvest loss and assessing their impact in these respective value chains. I'm a PhD candidate here at Iowa State University, and without further ado, I will start with my presentation. There we go. As an outline to this presentation, I will start by giving a background on the study that we conducted, and then I will list some objectives that we set out to accomplish. Then I will describe the method that we used to attain these objectives. I will show you the results that we obtained before concluding. So this study was initiated as an attempt to understand the impact of the Yieldwise initiative, which was launched in 2016. It was an initiative launched by the Rockefeller Foundation, and its objective was to help smallholder farmers reduce post-harvest loss in 3 commodity value chains that you see on this slide. It was tomato in Nigeria, mango in Kenya, and maize in Tanzania. So the Rockefeller Foundation worked in partnership with some implementing partners, and they provided some services, a number of services to the farmers, such as providing access to finance, training the farmers, and providing post-harvest technologies, and so on. After the implementation of those services, they then collected some data through a survey. And so that data set was made available to us. And what we decided to do with that data set was we came up with these 3 objectives. The first was, because there were so many variables in the data set, we decided to first identify the critical drivers of maize, mango, and tomato post-harvest loss. And the second objective was to assess the impact of the most critical drivers on post-harvest loss in each of these value chains. And the third objective was to establish an interactive web dashboard that will allow the user to visualize post-harvest loss as a function of the various variables that were in the data set, but also, and perhaps more importantly, to make some predictions of post-harvest loss by inserting a desired number of farmers that the user might be interested looking. So to attain these objectives, we decided to use a predictive modeling approach, and particularly the random forest predictive model, because the random forests create several predictive models called decision trees, and then it combines them together to produce a precise estimate that is low bias and low variance. So we found it very appropriate for this type of data sets. And so after identifying that, what we did was we went ahead for each and every value chain. We took the data set. We first partitioned the data set into a training set and a test set. And then we took the training set to build the random forest model. We checked the accuracy of the model. Then from there, we determined the variable importance. Now, this functionality of the random forest model was really important in attaining the first objective of our study, which was to determine the critical drivers of post-harvest loss. Then when we did that, we then modified the test sets, and then we could start making some predictions. And through the predictions, we created some partial dependence plots that I will talk about later in the results. And that's how we managed to assess the impact of the critical drivers on post-harvest loss. So when we did that, what we found was, in terms of the critical drivers of maize post-harvest loss, so this is the maize value chain, the quantity of harvest of maize that it was harvested by the farmers was the most critical driver of post-harvest loss. And this is just looking at the importance of the variable. But in order to understand the relationship between this quantity of harvest and the post-harvest loss, we created some partial dependence plots, as you can see here. And what the partial dependence plot shows us is that essentially when farmers are harvesting a small quantity of maize, typically less than 1,000 kilograms, then the post-harvest loss tend to be a little bit high. When they harvest a larger quantity, typically more than 1,000 kilograms of maize, post-harvest loss tend to be a little bit lower, regardless of whether there's 10 farmers, 50 or more. So again, but in addition to the quantity of maize being harvested, the production area, the geographic location of the farmer was also a critical driver, as well as the affiliation of the farmers to the Yieldwise initiative. In terms of the critical drivers in the mango value chain, what we found was that the way in which the farmers are being informed on when to start harvesting was the most critical driver of post-harvest loss. And so again, looking at the partial dependence plots of this particular driver, what we found was that there are 4 different levels for this critical driver. And when farmers are counting the number of days after the tree is blooming, and then they start harvesting based on that number of days, then the post-harvest loss is predicted to be lower. But when you start changing the number of farmers, you also see that there's a bit of an increase in post-harvest loss. However, that increase is not as pronounced as when you shift from one level of the critical driver to another level. So that's just something we've also observed with our results. And similarly to the previous value chain, what we see is the geographic location was also a critical driver, as well as some of the yield-wise initiative services that were provided to the farmers. And the web dashboard that we developed has more partial dependence plot for these other variables. And so the last value chain was the the tomato value chain in Nigeria. And what we found here in terms of the critical drivers of post-harvest loss, the pest attack, the type of pest that is attacking the fruits, the tomato fruit was the most critical driver. And looking more into detail at that, driver, what we found in terms of the partial dependence plot, that there were 3 levels, but the thrips bugs were the most, were predicted to have the least post-harvest loss. Again, similarly to the previous plot that we looked at, the shift in between the numbers of farmers is not as pronounced as when we move from one level of this driver to another. So with that, we actually came up with a few conclusions. So I will share that with you now. The conclusion is that the most critical drivers of post-harvest loss are mainly related to pre-harvest and harvest activities. And this is because, as you saw in the mango value chain, you could see that, how farmers are being formed to harvest mangoes was, typically a harvest type of activity. And also in the maize value chain, the quantity of maize that is being harvested, as well as in the tomato value chain, the type of pest attack. So all mainly related to harvest or pre-harvest activity. The affiliation of the farmers to the yield-wise initiative was a critical driver of post-harvest loss in all 3 value chains. Then increasing the number, and this we saw it in all 3 value chains again, increasing the number of farmers associated with a given driver has an impact of post-harvest loss, certainly. However, changing the level of a given driver has a higher impact on post-harvest loss. And perhaps the last conclusion was the fact that when you're trying to change post-harvest loss, it's important to remember that the critical drivers are both passive variables, or includes both passive and active variables. And what I mean by that is, for example, if you look at the geographic location, that is a variable that is very difficult to manipulate. It's difficult to tell a farmer, for example, to shift from one geographic location to go to another place so that they can have less post-harvest loss. And also, but at the same time, there were some active variables that were also critical drivers, such as the technologies that the farmers are using, and these are very easy to manipulate. So this is my presentation, and thank you for your attention. Thank you very much.
Great. Thank you very much, Ori. I appreciate it. We do have some time for questions here. We got 3 or 4 minutes. So if there are any questions, please let me know by putting something into the chat. I have a question, Ori. At the beginning, when you started this project, what were the biggest challenges that you faced trying to put this framework together to understand all the data that you had collected?
Yes, absolutely. So one of the biggest challenges was the fact that the data was collected by other organizations that I was not in contact with. So it was a little bit difficult in terms of using the right approach, because we did not know how the data were collected, We did not know the way in which they randomized their collections and in which they randomized the interventions of the Yield Rise Initiative, whether the farmers were selected randomly or based on a certain criteria. So all of those factors certainly had an impact on how we would go about doing the analysis of the data. And later on, the other challenge was the actual quality of the data, because there were quite a lot of missing values here and there. And so we had to interact with the people who were on the ground to understand exactly why those numbers were missing, or if sometimes there were some outliers, very large numbers. And so just looking at the quality of the data and understanding how the data was collected was certainly a big challenge.
So then the other question I had was, you use some perhaps less than familiar statistical techniques to analyze the data. Could you talk about how you arrived at that? I mean, as a researcher, you know, I'm used to using ANOVA and comparisons and all that kind of stuff, but you were doing things like that had names that until I saw your research, I had no idea existed. So can you talk about the process by which you got to that point to discover and use these statistical techniques?
Sure. So we use the random forest to do this analysis, and it's been used for various other applications as well. And the idea was really, we looked at the structure of the data, and there were quite a lot of variables, and statistic approach to understanding, especially when we're trying to identify critical drivers within a data set, they certainly can help, and they're really important, but that sometimes have their limitations. And the data that we had quite a lot of categorical variables. It was kind of a mixture of categorical variables and numerical variables. We had more than 100,000 data points. And so we wanted to make sure that we use an analysis tool that will be able to capture the complexity of the data while providing a very precise type of estimate. Since we're making predictions, so we wanted to use a tool that has very low bias, low variance, but at the same time, something that can capture the complexity of the data. So that's why we went for this.
Excellent. So there's a question here from our colleague in Rwanda, Kizito Nishimwe. Do you think these recommendations can be applied in other countries?
Yes, thank you, Dr. Nishimwe, for that question. So I think we have these can certainly be applied in other countries, because when we came up with these results, we actually looked at what the literature as also says about the critical drivers of post-harvest loss. And interestingly enough, we actually saw that, especially with the quantity of harvest, in what that we found in Tanzania, that's something that occurred in many other countries in Africa. So when, for example, a farmer harvests a large quantity, a small quantity of maize, then chances are they will have a bigger loss because of just not being able to have the appropriate storage technology. So small quantity of harvest is not really in the favor of smallholder farmers in Africa. And this is something that you see in many countries. And so looking at these recommendations and looking at the conclusions that we have can certainly help us in shaping how we go about addressing the issue of post-harvest loss, not only in these countries that we looked at, but in other sub-Saharan African countries.
One last question from the chat here. Can the presenter throw more light on how post-harvest losses can be minimal on a large farm rather than on a small size farm?
That's a very interesting question. I first have to say that before I answer the question, our study focused particularly on smallholder farmers. And what we mean by that is typically a farm that is not more than 5 acres when you think about especially with the maize fields in Tanzania and so on. And so if you think about large scale farmers, we haven't done much study in terms of that, but with what we've seen so far in the literature, most of the large scale farmers tend to have the infrastructure in place to already deal with and post-harvest loss. And I also have to specify the fact that post-harvest loss might not be a very big issue to those farmers. Their concern is mainly on the quality of the harvest and perhaps increasing the yield. But because they're producing very large quantity of crops, the percentage of loss tend to be very minimal, and it doesn't warrant much of their interest. So I would not. I don't think that large-scale farmers will be interested in so much in reducing post-harvest loss, or the benefit of post-harvest loss reduction will not be as important to them as it will be to the smallholder farmers.
So we do have another question in the chat. I'm going to save it. We have time at the end that we can go back to that. I want to make sure that we have plenty of time for the presenters. So thank you, Hori, for your presentation, the information that you presented. The next presenter is Christine Ignacio. She is a PhD candidate here in Ag and Biosystems Engineering. She's been working on hermetic bags, and she's going to discuss predicting the performance of hermetic storage bag technology. So with that, Christine.
Okay, thank you, Dr. Bram. Good morning, everyone. Good afternoon and good evening. I will present to you a part of my dissertation, which is the predicting the performance of hermetic storage bag technology. Hermetic storage bag technology works under the principle of biogenerated modified atmosphere, and it is achieved by the aerobic respiration of grains, insects, and molds. And with this, a low oxygen environment, it inhibits the development or the growth of insect pest, mold, and aerobic yeast, which are major sources of grain deterioration during storage. This technology is most widely disseminated among smallholder farmers in Central America, Asia, and Africa. It is chemical free because you don't need to use pesticides to prevent insect infestation, and it is easy to use. It has two components, the polypropylene bag as the outside bag and the plastic liner as the inner bag. The focus of my dissertation is the plastic liner. So I tested 6 different commercially available hermetic storage bags and determined their properties as basis for the initiative of ASABE in putting up a standard for this technology. Now, there are commercially available hermetic storage bags now, and they can be categorized into 3. Single layer, which consists of 1 inner liner. These are the super grain bag and the torso grain shell bags. And also the double layer composed of 2, which is the PP bag and the inner liner. These are the Agrosi bag and Cerafly bag. And also the triple layer, which includes 2 inner liners. And these are the Pix bag and Elite bag. Now, we can categorize them actually into 2 by considering the inner liner, and that is single liner. This liner is actually multi-layer films with low permeability barriers, and the other one is our double liner, which uses 2 HDPE liners. The impacts of hermetic storage bag technology has been established in many published articles and studies. And in social, it provides the farmers to the flexibility to store the grain for several months while waiting for prices to increase. So it's profitable, and it ensures food safety as different studies proven the efficacy of these bags in mitigating mycotoxin contamination. Economic, it reduces post-harvest losses due to insect pests, and also ensures food security for preservation of the quality of the stored products. Now, with the continuous demand for hermetic storage bags grow, more research is needed to address existing and emerging challenges. That includes, of course, the understanding the factors that influence achieving and maintaining a low oxygen environment. Through this study, we are predicting the performance of the hermetic storage bag technology by calculating the oxygen depletion at different scenarios using the grain respiration, insect respiration, and the hermetic bag liner property, oxygen transmission rate. The parameters used for the calculation of grain respiration are the following. We have maize at 50 kilogram, 13% moisture content at 30 degrees Celsius, and using the AACB dry matter loss equation. And the storage duration is at 90 days. Now for the insect respiration, the insect considered here is the maize weevils, which we have an average oxygen demand given in this table at 0.17 ml per day per insect and an average life cycle days of 36. In the calculation, in different scenarios, we use no infestation, low at 2 adult insect per kilogram grain, high at 20 adult insect per kilogram grain. And we consider 2 life cycle stages for the 90 days storage period. Now for the hermetic bag liner, we have a bag size of 70 liters. Exchange surface area for the transmission is 1.10 meters squared. And the types of liner that we'll be using is one with OTR value of 10 cubic centimeter per meter squared per day, and the other one is 50 cubic centimeter per meter squared per day. Now for the first scenario, this is with the liner with OTR value of 10. The oxygen depletion of stored grain at 13% moisture and 30 degrees Celsius to 5%. As you can see from the graph, reaching the 5%, we have the no infestation. It will take more than 90 days for it to reach 5%. And that is the no infestation is without the insect. And then for the low infestation, it will take 79 days, while for the high infestation, it will take 35 days. Now for the second scenario, using a liner with 50 cc per meter squared per day, the oxygen depletion of stored grain at 13%, 30 degrees Celsius to 5% is given in this figure. So for the OTR, high OTR value, you will see that our oxygen depletion until it's 90, it's still at 19%. That is no infestation. For the low infestation, we have around 16% at 90th day. But for high infestation, we have at 50-day period, it will reduce to 5%. Now, in summary, we have these different scenarios, different insect infestation, the days to reduce oxygen level to 5%. So if we have a lower OTR value than with no insect infestation, it will take longer than 90 days because the oxygen transmission, even if it's very low, the grain respiration here is higher, but still it will take 90th day to reach 66%. Now for low infestation, we have 79 days. Here, the insect respiration will kick in, and of course, there is a low oxygen transmission. And the high infestation, the reason why we have 35 days to reach to reduce to 5% oxygen level is because the insect respiration dominates the depletion. Now for the scenario 2, with no infestation, having a higher transmission, it is almost the same as the grain respiration. So meaning that is the reason why the oxygen level is still at 19% up to the 90th day. Now for low infestation, the oxygen transmission is higher, meaning the incoming oxygen is higher than the consumption, but still there will be depletion, but it will take more than 90 days to reduce to 5%. But for high infestation, so having the insect respiration and the grain respiration, adding them together as the consumption inside the grain, it will be greater than the oxygen transmission. So the reduction will only reach until 50 days for 5% to reach. Now, having that, we have that if we reduce the oxygen to 5%, that is with the assumption of killing of all adult insects. And if we maintain the oxygen level below 5%, then all emerging life stages from egg to larvae to adults will also be asphyxiated. So given the previous graph, we saw that only the low infestation, high infestation at low temp OTR value, and also at high infestation of high temperature, high OTR value will have the 5% within the 90 days. So we can see that after reaching the 5%, all the adults will be killed, and then we can see that it will continue to drop, even there are larvae and pupa stages of the insect inside the bag. Now for the summary, after reaching that, after oxygen level reduced to 5%, at this day, the reduction of oxygen level will continue until the 81 day, reaching 1% for the low infestation, for the high infestation, reaching 1% on the 30th day, and also for scenario 2, which uses liner to reduce oxygen level to 1% on the 54th day. So by these calculations and different scenario, we can conclude that at high infestation with low OTR liner results in less than 40 days to kill adults, and that is around 35 days, within only one life cycle of maize weevil. And the insect reproduction is suppressed, reaching below 1% oxygen level with additional of 4 days of storage. For low infestation requires more than 2 life cycles to get to 5% oxygen level, but the insect reproduction is suppressed with additional of 2 days of storage. If we have a high OTR liner, having a high infestation level, it takes more than 1 life cycle to drop below 5% oxygen level, that is on the 50th day, and it takes 2 life cycles to suppress the population completely. Insect reproduction is suppressed with additional of 4 days of storage. While if you only have low infestation level, it requires more than 3 life cycles to drop the oxygen level to 1% because the transmission is higher than the insect respiration. Now, for no infestation using 2 different liners, for low OTR, grain respiration causes the depletion of oxygen to 6% until the 98th day. While if you are having a high transmission of oxygen into the bag, your oxygen level remain above 19% after 90 days. With this, our final conclusion is a biomodified atmosphere in a hermetic storage bag changes as a function of insect infestation and oxygen transmission rate of liner, much more so than due to grain respiration at 13% moisture content. With these calculations and results, will ultimately allow for specifying the preferred application of hermetic storage bags with lower versus higher OTR values as a function of insect infestation and grain moisture content. And through this, after we finish all the calculation, we'll be able to develop an online dashboard for this for the public to use. Thank you very much. If you have questions, you may contact us on this information. Thank you.
Great. Thank you, Christine, for that. We have time for a couple of questions. If you have something, you can put it into the chat. I have a question. So you had 2 different kinds of OTR bags, 10 and 50. So yes. How would we know if someone were purchasing bags? What are those? I mean, are they labeled by the manufacturers? Are there particular types that have those levels? How would a purchaser of those bags know what the OTR level is?
Other hermetic bags liner commercially available, they have OTR value in their specifications, but not all the available hermetic bag liners. So that is the reason why first objective of my dissertation is to conduct this standard methods for determining OTR values for commercially available hermetic bag liners as basis for the development of ASABE standard on hermetic bags.
Great. I have a question here. Thank you. I work with rural women farmers and entrepreneurs here in Nigeria. Was Nigeria part of this study? Can the hermetic storage bags last effectively more than 90 days, maybe 1 year preservation efficacy without infestation? So how long do these bags last?
Okay, the claim of the manufacturers For the hermetic bags, it can be used for more than 3 seasons. So, or yes, more than 3 seasons of storage. So, but the question about 90 days, most of there are studies that they use the hermetic bag for storing our storing grain for more than 90 days. If you say no infestation, that there will be a question of if there will be no infestation, no adult insects initially, but if you have these eggs and then if you have a high value of more than 5% inside the bag oxygen level, it will continue the reproduction and will go on for a different life stages. And that is the reason why we're comparing the low OTR value versus the high OTR value, because it will differ on the oxygen coming inside the bag. If you have high OTR value, and then it will not really deplete the oxygen level inside, because the oxygen of consumption of the grain will almost be the same through time, because that's at a given temperature, at a given moisture content. The changes will only be on the how the bugs effectively control the transmission of oxygen, and also with the insect respiration to deplete to 5%.
Well, we have some more questions, but alas, we need to move on. We'll try to catch up to those at the end when we have some additional time. Our next speaker, thank you, Christine. Our next speaker is George Obang-Okrofi. He is a PhD student in Ag and Biosystems Engineering, and he's going to discuss the preserving the value of shea nuts for improved livelihoods. So with that, George.
All right, Professor Brom, thank you very much. Good morning, good afternoon, good evening, all our participants. My name is George, and I'll be presenting some work we've been doing in preserving the quality of shea nuts for rural livelihood. So with that, I'll move on. With the outline of my presentation, we'll touch a little background, talk on some research activity we've been carrying out, some deliverables and impacts we've made so far, and also highlights on some collaborative efforts we've had. So shea nut, as an oil-bearing crop, is produced in the wild of the Sahel region. So on the map on our right-hand side, you see the map of Africa, and the Sahel region is just beneath the Saharan desert, spanning from the western side up from Senegal way across to the eastern side, where we have countries like Ethiopia also having shea grown on their land. And as a fruit, it comes in this form, and in the transversal way, it has a fleshy fruit around it with the nut inside containing oil sources. And in Ghana, as of 2010, it was estimated that close to $200 million was generated from the exportation of shea butter and shea nuts. And as a woman-centered industry, it employs close to 6 million people in terms of women who pick the nuts, process the fruit into nuts, or even shea butter for household consumption. In order to assess how the challenges of this industry in terms of the post-harvest losses occurred, we decided to take out a baseline survey. So we worked in a community in Ghana. So on the map of Ghana here, you see on the Upper West region, we have a community there called the Ulo Traditional Area, which is just about an hour south of Burkina Faso. In that study, we surveyed 300 women in 7 different communities under the traditional area, and we had some focused discussions to also understand what some group of women also perceived in their challenges. Moving on in this study, we were able to find out that aside the demographics of the women in terms of their age, their working age, and level of education and all, we're able to identify that storage and drying continue to be a problem when it comes to shea nut handling in this community. So as you can see in this picture, it's a typical way by which these rural women tend to store their produce, where they have a closet, they have their shea nuts together with their bicycles and even some animals that they tend to cater for. Now, with some recommendation based on this survey that we did, adopting the yield-wise model, as my brother Harry spoke about in his presentation, we found out that introducing technologies, appropriate technologies to this industry will play a major role in improving the quality or maintaining the quality of the nuts. Also, access to training, aggregation, the importance of finance, and also linking the women to right market sources will tend to have a positive impact on their livelihood. On the issue of technology and access to technology and also training, what we did was to assess the efficacy of hermetic bags in storing these shea nuts. So at the rural level, as I mentioned, women move into the wild, pick up shea fruits, come back home and process them into shea nuts. And moving on, they tend to store the shea nuts either to process them into butter for cosmetic products or even for food at the household level, or tend to store them for better prices and sell them later for better value. However, due to these inappropriate storage measures, they tend to be faced with post-harvest losses, be it some qualitative losses or quantitative losses. So as shown in the picture up here is some shea nut that has been mold infested, and down here some shea nut that has been insect damaged due to some infestation of some Indian meal moth. So what we did was to carry out a 30-week shea nut storage in Ghana under an uncontrolled storage condition. So we had a simple warehouse on KNUST campus where we had 3 different storage treatments. So the pig's bag as a type of hermetic bag that we use, the polypropylene bag, and a jute sack. All these storage systems were actually done in the uncontrolled condition, as I said, but in the process, we also determined the temperature and relative humidity conditions in this bag. And after each sampling period too, we determined the carbon dioxide concentration in the pig's bag or under the hermetic storage, in addition to moisture content and insect damage or insect infestation. Now to the results. When it came to moisture content, we find out that when it comes to storing shea nuts under hermetic condition, there's a high potential to maintain moisture content variation over the storage period. And this is evidenced in the results that we show that for the nuts stored in pig's bag, there was just 1 percentage point difference or variation in terms of moisture content over the 30-week storage period. And this was not significant as the storage process went on. However, when we consider the jute sack and the polypropylene bag, there was high variations due to the weather fluctuations during the storage period. Up to 3% moisture content difference was witnessed. And this was really significant in terms of the weight of shea nuts across the storage period. Moving on to, we determine the percentage of weight loss due to the different treatment that we had. And we saw that storing shea nuts under hermetic condition also has a great potential to reduce the rate at which shea nut is being lost due to insect damage. And then again, for the hermetic bag, we could see that over the 30-week period, there was no significant difference between the nut that was initially stored and the nuts that we had after the 30-week storage period. However, the nuts stored in the jute sack and the polypropylene bag had some high, weight loss due to insect damage after the 30-week period, with percentage reaching 10% and close to 12% for the jute sack and also the PP bags, respectively. Now to the major conclusions that we had from this study was that hermetic storage has a potential for keeping the quality of shea nuts under a 30-week storage period under uncontrolled condition. And nuts stored in the hermetic bag had no evidence of mold development. And with all these quality preservation methods over the 30-week storage period, what we can say is that we reducing the rate at which shea nut quality is being lost is correlatively related to how much money these women can potentially save with the application or the use of hermetic bag in their handling of shea nuts. Looking ahead in this study too, we realized how important it will be for us to determine moisture content on a regular basis. So we are moving on with a study which will lead to developing the EMC relationship of shea nuts for monitoring nut quality during drying and storage. Now, equilibrium moisture content is actually the moisture content at which the internal water vapor of biological materials will be with the surrounding air, right? And this EMC has a high relationship to the drying and storage of any food products or grain or oil seed. So from the graph up here, we can see that for any biological material stored in an isothermic condition with constant temperature, we will have an equilibrium moisture content for these food products, right? So depending on the moisture content of the products, whether it's high or low, we will either have a desorption. In other words, when food product has high moisture content in a specific environmental condition, it will tend to release moisture until it gets to an equilibrium condition or equilibrium moisture content. And the vice relationship happens when you have a lower moisture content product, whereby the product will tend to absorb moisture to get to equilibrium. So applying this, what we seek to do is that we are going to put shea nuts in hermetically sealed jars, keep them in an environmental chamber with a constant temperature, and monitor the relative humidity condition in the jar until it reaches equilibrium. So we'll store shea nuts basically at different initial moisture content and see how these relationships varies across time. Once we get to this equilibrium, we develop an equation, and this equation, which will be a function of the moisture content, relative humidity, and temperature, will help us to determine the moisture content of shea nuts more effectively during drying and storage. Yeah. So when it comes to the main deliverables from this project, what we did was, after the 6-week storage period of shea nuts and seeing the good results that we had, we went back to the community and carried out a training session for the women in there. So what we did was, since we can't just focus on shea nuts, as in shea nuts being the only produce that they have in the community, we decided to train them on how to, maintain the quality of some products that they produce. So in the picture up here, you see ourselves giving them some flyers on how best to use the pig's bag. And the picture here is where we show the women the results from the study, letting them know the difference in quality of shea nuts stored under the different treatments that I spoke about. And yes, so as a result of these two, with our collaborators, Self-Help International from Ghana, we've been able to set up 2 local entrepreneurs to also be as some main dealers in post-harvest technology, considering the use of pig's bag and also some drying taps that can help the women to preserve the quality of their food products. And I can also move on without highlighting the importance of KNUST in terms of 2 students using various aspects of this project as their capstone project for their undergraduate studies. With all these deliverables that we've had so far, we also did that in the name of some collaborators that we have. So KNUST as a department or as a university in Ghana has been really helpful with helping us. Actually, during the start of this project, COVID hit, and so most of our travels got cancelled, but then we're able to team up with KNUST, work on this, most of our experiments together, and things turned out to be successful. For Self-Help International, yes, it's an Iowa-based NGO. They have an office in Ghana and focus in developing rural communities, and they have also been really helpful in terms of assessing like micro loans and other kind of things in rural communities. We also work with SoCheck, an Argentine company, an Argentine industry that develop carbon dioxide monitors and sensor technologies, which is also in Ghana, to develop some moisture meters that can help in determining the moisture content of food products in rural communities. All right, thank you for your attention. So if you have more questions, you can contact myself and my major professor, or you can use the QR code to go to our website and follow through with what we are doing so far. Thank you.
Great. Thank you so much, George. Appreciate that presentation. If you do have a question, we have about 10 minutes left that we can answer them. If you specifically have one for George or any of the other panelists, please go ahead and put them into the chat. George, can you tell me how big is the shea nut industry in Ghana? How widely planted are shea nuts and how important is it to the livelihoods of people there?
Yep. So when it comes to Ghana, the northern part of Ghana, which occupies about 1/3 of the country's area, is where shea nut grows. And it is interesting to know that in almost all these communities that grow shea nuts, the women in there tend to process the shea nut and use that as a source of food, like frying oil for food in there. So it's of high importance to them in terms of being a nutritional value crop and also a source of economic economic earnings to these women who trade the nuts and also trade the butter for economic gains.
Got it. I just want to remind everyone that this webinar is being recorded so that if you need to review it or some of it that you missed or you wish to pass the link on to others, and that'll be located on our consortium website. And George showed the link there in the presentation, but also if you look at the invitation for the webinar, it also has a link to that. So let me see, I'm scrolling through. Any collaboration with the private sector and Ghanaian investors to transform social economic lives of women in Nigeria in shea butter economy. So do you know anything about shea butter in Nigeria, George? Because I certainly don't.
Yeah, well, I'll say I've not widened my scope to the Nigerian economy yet, but for Ghana, yes, there are some investors such as the Global Shea Alliance and the Ghana Shea Network who are partnering with rural communities or rural shea growing communities and helping
She, in the chat, she meant Ghana, or meant Ghana, he or she meant Ghana, not Nigeria. So go ahead and talk about Ghana. Sorry.
Yes, please. Yes, please. Yeah. So as I was saying, there's the Global Shea Network and the Ghana Shea Global Shea Alliance, and the Ghana Shea Network in Ghana, who usually collaborates with women who are into shea processing and shea nut producing, and are helping them a lot in terms of linking them to some good market sources. Yeah.
Great. I have a question here for Christine. There is a perception that grains stored in hermetic bags are difficult to cook, that they take longer to cook or do not cook at all. Have you come across this claim in your studies?
Oh, no. From what I have studied or read, published articles are mostly on the grain assessment after the storage, and also published articles are into how many dead insects after the given storage, but not on the quality of the cooked grain stored in hermetic bags.
Yeah, and I can, I'm doing a lot of work with hermetic storage in Uganda, in particular with primary schools. We store all of their maize in hermetic storage, and we haven't seen any issues like that. So that might be a folk legend or some kind of rumor that propagates when we introduce new technology. Let's see, let me go back to, we had some questions for Hori. How could you, this is the work that he did with the YieldWise initiative. How could you communicate these outcomes to rural women farmers in Nigeria farming tomatoes and maize that suffer post-harvest loss? So Jorge, if you can maybe talk a little bit about what we could do with this information that we've determined.
Sure. So in the tomato value chain, the tomato value chain in Nigeria, as I presented earlier, one of the most critical driver that we found there was the type of pests that was attacking the fruits, tomato fruits. And one important mitigation to that would be, for example, being able to use some of the insecticide or pesticide that are on the market that are not very toxic, that could protect the crop, and also try to find a way to grow the plant into an environment that some people try to imitate the technology that people use, such as greenhouses, for example. They try to imitate greenhouses and try to do that so that it can mitigate the infestation of pests. So that's one approach, but there are also a number of studies that we're currently doing here in terms of coming up with a sustainable way of helping farmers mitigate post-harvest loss. And once we finish those studies, we will be able to get more recommendations as to how we can go about helping farmers there.
Great. And the other part of the question is, hang on, I just scrolled past it. What's the best technology that you can recommend to local farmers to mitigate these losses?
Wow, that is a very good question. So the best technology is not based on it's depend on what we're actually trying to achieve. A best technology, you have to look at the impact of a technology within the system and not just saying it reduces post-harvest loss. So the best technology will be one that not only reduces post-harvest loss, but allows the farmer to generate some income, but also allows to the community or the value chain actors to create some kind of manufacturing facility where they can produce those facilities. Because oftentimes what we see in many of these countries, Sub-Saharan African countries, the technologies are often imported. So there's a very limited access in terms of where it can be manufactured. And even when people have access to the technology, sometimes the maintenance of the technology can be very difficult. So really what we want to do is to get a technology that mitigates post-harvest loss, that is affordable to the farmer, that is not very harmful to the environment, and that actually has some kind of an impact on the social life of the farmer. So that would be the best technology. We're doing some studies to identify that, and we will be able to give more answer in that regard.
And just to acknowledge, this is a related question. How can we mitigate highly perishable products loss by using locally available materials, particularly in developing countries? I think you kind of addressed that already.
Absolutely. So we're currently working on a study that looks at that, and within the few months we will be showing our results. So that would be really interesting.
So here's a question for George. What is the right moisture for shea nut storage in hermetic bags?
Yeah, so the right, the safe storage of moisture content for shea nuts is 7%. Between 7 to 8% is what's recommended.
7 to 8% moisture content on a wet basis.
On a wet basis.
Yeah. Okay. If you have any other questions, I have one here. Jorge, could you just summarize quickly the main outcomes of your study?
So some of the deliverables of our study, one of them was we actually wrote a manuscript, so that is under review and it will be published soon. Another one is a dashboard that everyone can access. I will put a link, I will just share the link here in the chat, but it's a very simple dashboard that you can use. I will go ahead and just quickly show you within the last minute that we have. You have all the value chains that I was mentioning, presenting in my work. In this dashboard, you have the mango value chain, you have the maize value chain, tomato value chain. And what you can do just by clicking on the desired value chain, you can look at the post-harvest loss based on different variables that you might be interested in. You can visualize that. You can also look at, analyzing, looking at the critical drivers that I was talking about. The list, you see their importance in the left column. And then you can also look at some predictions. You have a prediction, a value over here of post-harvest loss. Then you can select the driver that you're interested in. And there are quite a number of them. And you can also change the number of farmers that you might be dealing with. And then you look at what the result of that prediction, of that scenario that you put in, what kind of prediction it gives you. So these are some of the deliverables that we've done. And as I said, I will go ahead and share the link to this dashboard so it can be available.
And will that link also be available on the Consortium website? That would be a good place.
We'll make sure that it is available on the Consortium website as well.
Got it. Well, golly, here we are at 9 o'clock. I want to be respectful of everyone's times. I'm sorry we didn't get to answer perhaps all the questions, but I want to thank the speakers, Jorge, George, Christine. Thank you so much for presenting your work. I'm fortunate that they're part of our research group and I get to hear all these things, and it's great. We're grateful that we have this opportunity to share their research with you. Again, this is just one of a monthly series of webinars sponsored by the Consortium. So stay tuned for an announcement for the next month's seminar. Again, you can find everything, recordings, other information posted on the Consortium webinar or on the website. So thank you, everyone. Appreciate your attendance, your attention, and have either a good day or a good evening. Thank you so much.